All right, welcome everyone. Thank you for coming to my colloquium presentation. Of course, for those of you who do know me, you know my name. But for those of you who don't, my name is Thornton Raskevitz. I'm a master's student here at Oklahoma State University, and I will be presenting my thesis here during this colloquium presentation. All right, so getting right to it. So Pahytolith analysis as a paleoecological proxy when examining bison anatomical and behavioral changes in the Great Plains. So the order of this presentation, it will be, I'll go over a problem statement, I'll give a little bit of background, we'll talk about the study areas that I covered, we'll talk about the methods I use, then we'll discuss the results, we'll do a discussion, conclusion, and then finish up with discussing how we're going to go about creating a forum for questions. Alright, so what I tried to do with this project was examine Pahytolith analysis, especially dental Pahytolith analysis, as a tool for helping understand bison paleoecology, specifically focusing on the period between the kind of beginning of the Younger Dryas period at 12,850-ish calibrated years before present, running through kind of just over the end of the Younger Dryas, and then comparing them to modern references. And we thought that Pahytolith analysis would work pretty well for this because, of course, bison rely on the consumption of grasses for their daily metabolic intake, and these grasses are the dominant producers of phytoliths within the Great Plains ecosystem. So, a little bit of background. What do we know? Well, for one, we know that the bison changed. The bison that existed during the Younger Dryas were much larger than the ones we have today. There may also have been other anatomical and behavioral changes that could have gone along with that, but those are much harder to track. Size is pretty easy, we can use physical records, but for those sort of associated behavioral changes, we have to use proxy records and kind of suss out what they can tell us about the process of diminution, which is the process of getting smaller through time, and these behavioral changes that might have gone hand in hand with that. To understand this, diet is really important because diet is how an individual animal interacts directly with its environment. So it's almost impossible to overstate the importance of diet when discussing behavioral inputs on a population. The only thing that even comes close is, sexual selection, things like that. So what I did in this process, as I compared populations of Bison antiquus and Bison occidentalis to modern references from bison through the lens of Pahytolith analysis, especially dental Pahytolith analysis. So we know these bison changed. How did they change? Well, the three species that I looked at were Bison antiquus, which were taller and heavier built than modern bison. Their weight was 802 plus or minus 183 kilograms. These values are quoted from Martin, Mead, and Barboza 2018. These weight estimations on all of these are going to be based on the dimensions of the calcaneum, which is the bone in the heel. Second species, Bison occidentalis. This is somewhere in between Bison antiquus and the modern bison, with a weight of 678 plus or minus 105, again based upon the calcaneum. Modern bison weigh 479 plus or minus 177 kilograms. In the paper they had estimations based upon the calcaneum as well as direct measurements. So here's just a figure I made up to illustrate this size difference. We have bison on your left, and then Bison antiquus here on the right. As we can see, it was much, much larger in the ancient bison than the modern one, roughly 25%. in terms of height at the shoulder. And the calcaneum is this little bone here, or calcaneus. Both pronunciations are correct. So, bison as a study system. Bison were a good system to kind of look at this in because number one, they're ecologically important. They're the keystone species of the Great Plains ecosystem after that post-terminal Pleistocene megafaunal extinction event. Secondly, archaeological specimens are available for study. While they're not necessarily common, the remains are widespread in the Great Plains, and because of this, I had access to some to do this research on. Thirdly, as we discussed before, bison are obligate grazers. They feed on these grasses, which are the dominant producers of phytoliths within this ecosystem. So, you've heard me say this term phytoliths several times before, but what are phytoliths? Well, phytoliths are small, siliceous infillings of cells. So the cells deposit. Yeah, sure. You can zoom if you'd like, if you think that would be helpful. They're small, very small. We're talking, 20 micrometers, roughly. Deposits of silica within the cells themselves. There are three main types. These are the trichomes, the long cells, and the short cells. The trichomes, I have a picture of one here in G, are these kind of long, spiky looking things, and these are thought to be deposited for protection, especially for microherbivores, and pathologic fungi. They're thought to help protect against them. And then we have the long cell. I have two different long cells. B is a elongate, and then C is a rectangular bulliform. And these are thought to be deposited for aid in terms of support structure. And then thirdly, we have the short cells. And these short cells are important because these are the ones that are actually diagnostic. So we can, depending upon what type of short cell morphotypes we find, and we can find all of our short cell morphotypes here in figure A, we can determine what family or subfamily of grasses that those came from. So in this study, we focused on the Poetia grasses, which we have the keeled, pyramidal, conical, round trapezoids, the long wavy trapezoids, and then the trapezoidal bilobates. Then the Panicoidea, which are panicoid bilobates or dumbbells, the crosses, and then the polylobates. We have the Chlorodoidea, which are the short saddles, and then the Aristodoidea, which are long shank bilobates. So here in the photographs, we have a round rondelle here, which is a Poetia phytolith. We have a panicoid bilobate here for E, and then in F we actually have two Chlorodoidea short saddles. So, number one, these phytoliths can become deposited within the samples that we looked at in several different ways. The first type of sample, of course, was the dental sample, dental phytolith analysis. They become deposited within these dental samples due to the mastication process. So as the bison chew, these small siliceous structures are again hard. They actually become impacted in the plaque on the teeth, or the calculus on the teeth. And then within the soil, it's a little bit easier. Again, the plant, which holds the phytoliths, it dies, rots away. Because of their non-biologic siliceous makeup, these phytoliths are left behind. These phytoliths are useful as a proxy measurement because, number one, they're very long enduring. Because, again, they're very small structures. They're fairly sturdy. There is, within the literature, a reference to a diagnostic phytolith found within a Maastrichtian-age dinosaur coprolite. So we're talking in the span of hundreds of millions of years old. Of course, this is by far the exception, but it is, again, relatively common to find them in samples tens of thousands of years old. And then secondly, as we talked about before, they're diagnostic. So depending upon what we find, we can tell what existed in these areas or what these animals might have been chewing on. In this study, as well as in other phytolith research, phytoliths are often used in indices. The two that I use in my research are IPH, or the humidity aridity index, which is the Chlorodoidea phytoliths divided by the sum of the Chlorodoidea plus the Panacoidea phytoliths. Higher index values are going to indicate drier climates, and that's because the Chlorodoidea grasses thrive in xeric environments. Then I use the IC index, or the climatic index, and these are the Poidea phytoliths divided by the sum of the Poidea, Chlorodoidea, and Panacoidea phytoliths. Higher values here are going to indicate a cooler climate or more available moisture. So, my study areas. I have 4 modern sites and 5 different archaeological sites. My modern sites are the Niobrara Valley Preserve up here in northern Nebraska. We have the Sand Sage Bison Range here in western Kansas. We have the Tallgrass Prairie Preserve here in northern Oklahoma. And as well, we have the Lake Scott State Park in western Kansas. Lake Scott State Park, I don't have dental phytolith values from there because there is no modern bison herd on that landscape. For my prehistoric sites, we have 5 in a east-west transect here. In terms of oldest to youngest, we have the Jake Bluff site here located in western Oklahoma. We have the Cooper site here. Again, this is only located a couple hundred yards from the Jake Bluff site. We then have the Folsom site here in northeastern New Mexico. The Badger Hole site, which again is right next to the Cooper and Jake Bluff. And then we have the Ravenscroft 2 site, which is the youngest of my prehistoric sites here within the Oklahoma Panhandle. So, for my modern reference sites, the Niobrara Valley Preserve, I have both soil and dental values from here. This is dominated by Nebraska Sandhill Prairie with mixed grass prairies and tallgrass prairie. Tallgrass prairie occurs along the floodplains. Mixed grass prairie occurs in the highlands, or in Nebraska Sandhill Prairie kind of exists in those areas that are in between. The average yearly temperature is 9 degrees Celsius, and it receives 88 centimeters per annum of precipitation. The Tallgrass Prairie Preserve, again, I have both soil and dental values. This is a tallgrass prairie dominated ecosystem, but it does have areas of mixed grass prairie in areas that experience a high browsing pressure, as well as areas with very little soil, or sallow soil depth. You get that mixed grass prairie. The average temperature is 15 degrees Celsius, and it gets 89 centimeters per annum of precipitation. The Sand Sage Bison Range, again, soil and dental values. This is a sand sagebrush prairie. Our average yearly temperature is 12 degrees Celsius, and it receives 64 centimeters per annum of precipitation. So it's significantly more xeric here in Sand Sage. And then Lake Scott State Park, this, we only have soil values from here, but it was included because it is a very typical short grass prairie system. There's also mixed grass prairie and aquatics located right near the lake. The average temperature is 12 degrees Celsius, and it receives 56 centimeters per annum of precipitation. So it's the driest, and then second driest, third, and then fourth. So, my archaeological sites. Jake Bluff site is the oldest. It's dated to 12,712 plus or minus 122 calibrated years before present. The bison species that exists here is Bison antiquus. It is actually Clovis age. The bison were killed here during the fall. It is an arroyo kill site. There was a minimum of 22 individuals killed there. and I had access to samples from 2 different individuals. The second site was Cooper. Cooper dates to 12,569 plus or minus 47 calibrated years before present. This is the middle Cooper site. There are actually 3 Cooper sites that all lay one on top of each other, and they're all within about 100 years of each other. This again has bison antiquus. It's of Folsom age. The bison were killed here during the late summer or early fall. Again, it's an arroyo kill site, so the bison were driven in to this blind arroyo where they couldn't climb the sides, and then they were speared from up atop the bluffs by a cooperative group of hunters. There are 3 distinct bone beds. There were, in each of the bone beds, there was 20, 29, and then 29 minimum individual kills. I had a maximum of 7 samples, individuals sampled. I don't actually know the exact number because the teeth that I had access to were actually from the slump deposit, so they were bones that had fallen out of the bluff, and so they didn't have a specific individual that they were attributed to. So I know I had access to 7 teeth. They probably didn't come from the same individuals, but they may have. So a maximum of 7 individuals sampled. The Folsom site, this is the type site for the Folsom cultural complex. It dates to 12,450 plus or minus 86 calibrated years before present. Again, this is a fall kill, again in the arroyo kill style. A minimum of 32 individuals killed with 5 individual samples. The samples that I had access to were from the Figgins excavation in 1928 or 6. It was either one of those 2. The 4th site was the Badger Hole site. This dates to 12,145 plus or minus 79 calibrated years before present. This was, again, bison antiquus. This is late Folsom. The bison were killed here in the late summer or early fall, as is the pattern. Again, arroyo kill site, 20 plus individuals killed. I only had access to 1 individual to sample from here. And then my 5th site is the youngest. This is the Ravenscroft 2 site. It dates to 10,545 plus or minus 49 calibrated years before present. This is after the end of the Younger Dryas climatic event. We actually have that intermediate species, the Bison occidentalis, here. It's Paleo-Indian in age. The individuals killed here were killed during the winter. It's a very large kill site with 90 plus individuals killed, and I had again access to a maximum of 5 individuals, again with unassociated teeth samples. So, my methods. There were 2 different types of phytolith data that I obtained for this study. The first of which was soil data. I had soil data from all of my modern references, and then I had dental samples obtained from the teeth, dental samples. These were from Niobrara, tallgrass, sand sage, as well as all of my prehistoric samples. The soil samples were already prepared. I just processed them for phytoliths, so all I did was went through and counted them, and then each of the dental calculus samples had to be prepared before I could process them, and then process them and determined what they had for phytolith within them. So, sampling the teeth. When you go to sample a tooth like this is a molar from the Cooper site. Each tooth is gently washed in water. It's distilled water, and you then gently brush it. You do this to get rid of any dirt because you don't want any phytoliths from the soil that is on the teeth to disturb your dental sample. You don't want any contamination that way. Calculus is then scraped off the face of the tooth with essentially just a dental tool. You know, same thing you use or you have used on you when you go to the dentist's office. The extracted calculus is then weighed, and then the extracted calculus is digested in an acid bath. Calculus is made up of calcium carbonate, CaCO3. So we put it in some dilute HCl. The HCl then dissolves that calcium carbonate, and what's left is the stuff that was stuck within the calculus itself. So this is any dirt that's stuck in there, this is any phytoliths that are stuck in there, any other things. To then reduce the acidity to neutral, we can't put a basic in it because that may damage things in there. So what we do is we actually decant the sample and add distilled water to reduce that pH. And we do that a minimum of 7 times to reduce that pH down to neutral. And then once we're done with that, we check that it's neutral via pH strip. We then decant it a final time and then pipette what is left onto a cover slip, and then we let that cover slip dry. We then mount that cover slip onto a slide using a heavy and telling mounting medium, which gives us a very permanent and clear slide to do our observations through. So the next step, of course, was microscope work. This was very time consuming. The prepared slides are scanned fully at 400 times magnification. The phytoliths are observed, identified, and then tallied. So you keep a tally of how many of each type of phytolith you've seen per slide. Each slide was scanned in a grid pattern, an overlapping grid pattern. I always started in the top right for whatever reason. You then go across, down, across, with a little bit of overlap at both the top and bottoms to ensure full coverage of the slides. So, results. What did I find? Within the Niobrara samples, I had 405 diagnostic phytoliths. In the soil sample, 638 diagnostic phytoliths within the dental sample. Tall grass, again, very similar numbers, 422 within the soil, 517 within the dental sample. Sand sage, 380 within the soil sample, 166 within the dental sample. And then at Lake Scott, I had 305 within that soil sample. My archaeological sites, my totals were a little bit depressed from that because, of course, not every phytolith survives deposition. At Jake Bluff, I had a total of 22 diagnostic phytoliths from that sample. At Cooper, because I had more individuals to sample from Cooper and they were of relatively high quality, I obtained 144 diagnostic phytoliths from the Cooper samples. From Folsom, I had 41 diagnostic phytoliths. Badger Hole, 31, and then Ravenscroft 2, I had 43. So, this is a graph that I suppose is probably pretty hard to see on the video. It just gives you the percent of the total phytoliths by type in each of my samples, starting with the soil samples here on top, then the modern dental samples down to here, and then these are my archaeological site samples there. The Poidia phytoliths are in the blue, the Panacoidia phytoliths are in the dark green, the Chlorodoidea phytoliths are in the light green, and the Aristodoidea phytoliths, I had very few of them. The maximum total was, I think, less than 5%, and these are in the yellow. I then have some of my non-diagnostic short cells in the orange. This is the corresponding graph. This shows my other type, my elongates, my trichomes, and my buliforms by percent phytolith total. I then have Poidia as a percentage of short cell for my soil values, modern dental values, and then my ancient dental values. Notice that the ancient dental values are all quite a bit higher than any of my modern references. Panacoidea is a percentage of short cell in the dark green. Again, notice my low values for panacoids on my ancient sites. Chlorodoidea is a percentage of short cell. Again, see, I've got high values here. I also have high values for Cooper as well. And again, Aristodoidea, you're talking very low percentages. And then my two results for my Pahytolith indices, my IPH aridity index and my IC climatic index, and those results will be talked about a little bit later. So here I plotted the aridity, the total for the aridity index over the site's total for the climatic index. And what we can see is almost four groups, well, three groups kind of forming. So we've got the ancient sites up here. We have the Cooper, which is going to pair pretty closely throughout this research with the sand-sage dental samples, as well as the sand-sage soil and the Lake Scott soil in the, kind of the second group on that second vertice of the triangle formed. And then you have the other two modern sites, Tallgrass Dental and the Niobrara soils. They're over there on that third vertice. I think I went one too far. Here we go. All right. Then I performed a cluster analysis. As we can see on the cluster analysis, two large groups are formed. These are the modern plus Cooper group and the archaeological group. And within the modern plus Cooper, we can also see, again, Niobrara and Tallgrass grouping together. Cooper pairs with the sand-sage dental values because of that high proportion seen within the Cooper sample of the Chlorodoidea phytoliths, and to a lesser extent, the relatively high sample of Panacoidea phytoliths. The physical or temporal location of each site seems to have had quite little effect on clustering within the ancient samples. However, sample type does seem to have an effect on clustering. As we can see within the modern samples, again, generally dental samples are going to cluster with other dental samples, and then soil samples are clustering with other soil samples. The one thing that does seem to make some sort of a difference, at least within the modern group, is the amount of precipitation. Remember that Tallgrass and Niobrara had very similar precipitation amounts, and they grouped together, as well as Sand-Sage and Lake Scott had very similar precipitation amounts, and they grouped together. So that's an interesting thing to note. All right, and then to try and determine why Cooper was behaving differently than all of the other archaeological samples, I performed a clustering based upon the totals of each individual sample that I had. This came out okay. It's probably really hard to see here. I chose not to include any samples that had less than 15 total phytoliths, because what I found was that they were just grouping by themselves regardless, because of one phytolith one way or the other made too much of a difference within the sample. So what we can see here, or determine here, is really one interesting thing. Is we can see that the Cooper samples are really displaying kind of a bimodality. We have three of our Cooper samples which group with the modern Sand-Sage samples over here, and then we have three of the Cooper samples which group with our other archaeological sites over here. And we'll discuss why we think that may be happening a bit later. Then I performed a principal component analysis. These are my plots for component 1 and 2, which together explained it was about 96% of the sample variation. So component 1 is defined positively. Component 1 is this axis here, defined positively by the number of Chlorodoidea and negatively by the number of Pahytolith phytoliths over there. Component 2 is defined positively by the panacoids and negatively again by these Pahytolith, as well as a little bit by my Chlorodoidea phytoliths. So again, what we can see here on how our individual sites score on our PCA is that these same three groups seem to be formed. We have the Niobrara and tallgrass samples, we have the Lake Scott, Sand Sage, and Cooper samples, and then the rest of our archaeological samples over here. So, discussion. The values that we see from the Pahytolith indices of all of the ancient sites that we sampled, Cooper appears to be the driest of them. However, it's not as dry as Lake Scott or Sand Sage within the modern reference samples. All of my archaeological samples actually fit fairly neatly between the IPH values that were returned for Sand Sage and the IPH values that were returned for tallgrass. So right in between those two sat all 5 of my archaeological sites. For the IC, again, all ancient sites scored as cooler than all of the modern sites. Cooper scored as the warmest of that group, which, given its position within the Younger Dryas climatic event, is a little bit anomalous. This may be due to the large proportion of Chlorodordia within that sample, which would have diluted the proportion of Poedia phytoliths recovered. This universally lower rating in my archaeological sites may have been influenced by changing dietary preferences, because bison at these sites may have selected for the softer Poedia grasses over the tougher C4 grasses. There is evidence within the literature that they may have done that. We also can see that in the values that we got for the PCA, with all of the modern samples doing a little bit of that, scoring lower on that component 1 than the dental samples scored lower on component 1 than any of their corresponding soil samples. So, the clustering of the samples indicates that Cooper notwithstanding, the bison at the ancient sites in general fed on a different proportion of grasses than are available at any of the modern references. Cooper bison may have fed on a mixed grass prairie similar to what is found at Sand Sage. This is based upon the clustering of Cooper with Sand Sage and the scores for my climatic indices, which scored similar again to that of Sand Sage. This may have been produced by the drier conditions during the middle of the Younger Dryas climatic event. There is evidence in my PCA analysis that the modern bison selected slightly against eating the Chlorodordia grasses, which are a little bit tougher, when other forage is available. Again, there's evidence for this in the literature and in the ancient bison, and my results cannot dispute those findings. So, in conclusion, the bison diet that we observed has changed between the two periods. The bison that existed in my ancient sites were eating different things than the bison at my modern references. One of the leading hypotheses for diminution, again that getting smaller over time, is that it occurred because of changing grassland composition. And my results here, again, because we can